certified in family practice. I've been in practice since 2001. I've got 31 years since the time I was started pre-med until now without any gap. I've never had more than a week or two a year off. In fact, I haven't had a real vacation in probably 15 years, which is something I'm trying to figure out how to make that happen. But I'm also passionate about what I do. And all I can say is it's keep learning. And this information, like I said before, I always tell people I'm a big fan of Dr. Halasa. I don't say that the sound like appropriate. I say it because it's true. Dr. Halasa is a true expert. People may not like it when he speaks his mind and tells people the truth and corrects them, but he really does understand the molecular mechanisms behind what's going on in the body. It's so deep, much deeper than most people. I haven't come across anybody that knows this science better than him. And maybe some people don't like the fact, maybe they want to be right. Maybe he doesn't say everything that's mainstream or what the standard of care wants us to believe, but I believe in the truth, and that's how I am. So I know that Dr. Halasa speaks the truth. He doesn't say anything without backing up what he says. And for that reason, I'm excited to hear what he has to say today.
Thank you very much, Dr. Joseph. Of course, you're my buddy, so you'll say good things about me.
Well, I mean, we became buddies naturally, the way it happens, you know.
Right.
I can tell that you're speaking on things that need to be heard, and you're also providing solutions that are making a difference. Even if you're behind the scenes I'm more in the clinical arena, the results downstream are real. And there are people right now whose lives have been impacted dramatically because of the things I've learned from you. So that's what it is. And you're a good friend.
Well, we're learning from it. We're learning from each other. I don't like to be in a position, we're all doctors here, even when I attend the conference of the Environmental Medicine Academy of Environmental Medicine. I see myself and all other people there, they are learning from each other. It's not a teaching, supposedly it's not to be a teaching session. It's more of discussion between doctors and trying to benefit from each other, from our and share our experience. And I'm open for anyone any one of you guys to you know, kind of correct me if I'm wrong. It's okay. I like that. You can cut me off at any time. I don't see it as interruption. I see it. It's way of interactive learning. So feel free, if you are losing the chain of following up with the lecture, please go ahead and ask questions so we can have better understanding and clarification. Because you know, it's all about the audience and how they perceive the information, and sometimes you know, the information is not well being broken down and processed. So the audience needs to help for that, so they can follow up. Otherwise, they will be sleeping. And I don't like to have a sleeping audience. I like to have interactive audience, so I can learn from you, all of you. And then from your side is to come up with your own lectures and expand what we are learning together. That's very important too. So I want to learn from you as much as you know, you want to learn from me and from others.
One little thing, if you don't mind.
Yes, go ahead. Yes.
I was going to say, you know, one of the things that comes up is, well, what about, you know, what about these other fields? What about acupuncture? What about meridians? What about, that's fine. From my perspective, there's a whole spectrum of things, you know, that are important. But my interpretation of what you're teaching is very specific and orderly. in the realm of molecular biology and the things that that are supported by the data, if translated correctly, to prove this point. It doesn't rule out meridians, acupuncture, yoga, spirituality. That doesn't mean that that's not important. It's just that this is very specific to a process that would be very valuable for physicians to challenge their understanding of, because this is what's happening to us more and more directly with what's going on today. That's it.
Sure.
I'm good.
So let's start with the title of this lecture, which is Unlocking the Power of Redox, Metabolic, Immune, and Targeted Biological Nanomedicine to Prevent and Manage Chronic Disease, including cancers and COVID-19. It's going to be a very long night, a lot of chemistry. So I want you to be kind of prepared. And if you have a coffee, that would be nice, because there will be a lot of chemistry and pathophysiology and connecting the dots here. And yes, in order to manage chronic disease, you have to restore the redox balance. So you have to understand the redox codes. You have to restore the metabolic balance. So you have to understand the metabolic codes. You have to restore the immune balance. So you have to understand the immune code. And also you have to deliver the drug to the targeted or to the pathology using nanomedicine. I think it's the best way, because we find out naked therapies, which all this injectable that we do, when you inject it IV, 0.01% gets into the target pathology. But once you nano it, it goes up to 1%. And if you use biological autologous, you may go more than 1% to the pathology. And then you can activate those drugs, those antibiotics, those cancer drugs with light, and that would generate free radicals, and that will induce apoptosis and killing the cancer and the pathogens. And you can break the resistance to the chemotherapy or to the antibiotics. Okay, and so let's start with understanding how your immune system works, the immune codes. Let's unlock the immune codes and how the innate immunity works with acquired immunity in shaping the immune defense against the invaders, against the pathogens. And so really what determined the direction of the immune system is the macrophage and what the macrophage eats. So it's all about the food that's being introduced to the macrophage. And then the macrophage will be secreting cytokines that will determine the polarization of your immune system, including the innate immune system. So let's say the macrophages ate infected cells with virus, or ate cancer cells. What will happen to those macrophages, those naive macrophages or dendritic cells? They will polarize to M1 or DC1, which is macrophages type 1, and they will start secreting interferon gamma and interleukin 12, and this will polarize the naive T cells into Th1. And then the Th1 will start activating naive cytotoxic T cells to become active cytotoxic T cells, and then active cytotoxic T cells will start attaching to those infected cells with viruses or cancer and release those cytotoxins and blow them up. So that's the mechanism of how your immune system is polarizing the acquired and innate immunity to destroy cancer cells and infected cells with viruses. So this is how we defend ourselves, how the immune system defends us against intracellular pathogens.
Dr. Halasa,
Yes.
do you want us to ask a question while you are doing it?
It's up to you. I have no problem. You want to ask questions during the talk, after the talk?
Well, that will probably emphasize what you are talking about.
No problem, Dr. Patel. Yes.
When you mentioned about that innate immune response and the macrophage plays a great role, but it is very important to understand that macrophage are there. They may be active macrophage or they be non-activated sitting macrophage doing nothing. Number one. Number two, that there are two types of macrophage. One is pro-inflammatory and one is anti-inflammatory. So it is important to mention. And then the when macrophages are not working optimally, then their cascade of reaction occurs. And cascade of reaction would be, just as you mentioned, that activated macrophages, IL-12 and polarization of naive cells to Th1 and naive cells to cytotoxic T cells and so on. But I think it is very important factor to know that we are talking about activated macrophage and not the macrophage which is just sitting there in extracellular matrix doing nothing.
Right. So we're talking about what polarized the immune system into certain subsets of lymphocytes, and why, when you are being infected with viruses, why we have lymphocytosis versus when you are infected with bacteria, you would have neutrophilia. So we want to try to unlock this mystery by starting with ABC. Of course, there's a lot of variable there, and we're going to talk about them, but at least we can have some basic language that we can understand and agree. And then definitely there's a lot of other things involved. In biology, there's a lot of exceptions there. So the other thing, if the macrophages ate bacteria or ate fungus, this macrophages will be polarized and become macrophage type 2. And then they start secreting all this interleukin-4 and polarize the lymphocytes to activate or become Th2, and then those naive T cells become Th2, and then Th2 will start activating the plasma cells to secrete immunoglobulin E, and then this is how your body responds. Sorry, we said bacteria. Sorry. So in case of bacteria, when you are eating bacteria, the macrophage eats bacteria, it polarized the immune system to Th17. Sorry, not Th2. It polarized the immune system to Th17, and the macrophage become macrophage type 17. And then those naive T cells, they become Th17, and they start secreting interleukin-7, interleukin-6, and interleukin-6 polarize the CBC into neutrophils. So you see the response here is neutrophilia, and that's what the proper response to attack the bacteria and fungus. But in case if your macrophages eats allergens or parasites, then your macrophage will polarize into M2, yes, and then the M2 will start secreting interleukin-4. Interleukin-4 polarize the naive T cells of the Th2, and then they start activating the plasma cells to secrete immunoglobulin E, and that's what you see in allergic reaction and parasites. You will see immunoglobulin E attack against those parasites, and you will see eosinophilia and basophilia. See how the immune system is polarizing the acquired and innate immune system at the same time. And so when you do CBC differential, you will see eosinophilia and basophilia when the body is invaded by a parasite, or are you allergic to certain things like pollens, and you are encountering pollens, you will see that the whole immune system is polarizing into Th2. When you see here that we are the macrophages eats bacteria or fungus, the polarization moves to Th17 and to neutrophilia. That's what you do CBC differential. You see neutrophilia, and when we at the immune system is or the macrophages is eating cancer cells or cell that's being infected with virus, the polarization will be toward Th1 by secreting interleukin-12 and gamma. So this cytokines really helps to direct the immune system, the acquired and the innate immune system. And that's the reason we order CBC differential. And when we see that, oh, there is neutrophilia, so that's mean, oh, this could be bacterial infection or fungus. Oh, if it's basophilia and eosinophilia, oh, this could be allergic or parasite. And so we're expecting there will be too much of Th2. And when we have bacteria, we're expecting too much of Th17 here, interleukin 6. And when we have cancer or virus, polarization is to lymphocytes, lymphocytosis. So this could be cancer or virus. And then we start doing all this lab work to figure out whether it's cancer or it is virus. And then we also, when we have eosinophilia and basophilia, is it this allergen or parasites? But what about the body when they start eating your own self antigens? Then what would happen? Those macrophages, they polarize into Treg macrophages, Treg dendritic cells, and they start secreting all this transforming growth factor interleukin 10, which polarize the Th naive cells to Tregs. And Tregs, when they secrete interleukin 10 suppress all the immune effectors out there, the Th1, the Th2, the Th17. This is the language. This is the codes of the immune system, the basic codes that we need to learn, because then we can understand where the direction of the immune system just by ordering CBC differential. So now that CBC and differential is very valuable test, because it will tell me if this is immune response is Th1 dominant or Th17 dominant or Th2 dominant. But of course, as Dr. Patel is saying, there's a lot of variables there. You could be having Th1 and Th17 dominant in one disease like multiple sclerosis. You can have Th2 and Th17 dominant in COVID-19 virus. And that's a little bit, there's exceptions there too, because all the viruses, when we get infected with all kind of viruses, most of the polarization is to Th1 in lymphocytosis, except COVID. The polarization is completely different. And that's why biology is also different. And COVID, the polarization is to Th17, Th2. And we're going to learn why COVID virus is exception, is different from all other viruses. That's why in the beginning, when we start discovering about the COVID, and we saw, hey, there's neutropenia, so it must be some bacteria, or it could be the secondary infection is causing neutropenia, not the virus. The reality is that COVID-19 is the reason of polarization to neutropenia, which is completely different from the basic that we learned. We learned that virus has to move to Th1, not to Th17. But COVID, for some reason, polarized the immune system away from Th1, which is something not good, because then we cannot fight the virus in the right way. And that's why this virus is very unique in hijacking the RAS system. And that's the reason why this virus is polarizing the immune system into Th17, Th2 in a state of the right way, which is Th1. Because if it's polarized Th1, that would be great. We can get rid from the COVID. But polarizing it away gives us a hard time to get rid from COVID. And we're going to study that and learn that and come up exactly the reason why this virus is different from all other viruses. Are you there, guys? Can you hear me?
Yeah, we can hear you. Great. Just really quick, just the box that we see you in is blocking the Th1 response. If we could just really quick, what's
oh, the box, that there's a box there too?
No, just where you're speaking, where we see you speaking is in the upper hand, right hand part of the screen, and it's blocking what Th1 is causing and producing. And I just don't want to miss a piece of this beautiful puzzle here.
Right, and here we go. Intracellular bacteria, anything intracellular, yes, pathogens, bacteria, and virus, you will get the Th1 response. But extracellular bacteria, you will get the Th17, and including the cancer. It moves to this direction to Th1. And then Th2, you see here, extracellular parasite, allergens, and then Th17 is the bacteria and the fungus. And Tregs is the way that we can suppress the immune system and produce what we call tolerance, right? And prevent the immune system from attacking your self antigen. But there's other things involved as well. Make sure, please add people, because I will be lecturing and I've been missing people who are being in the call. Now, let's go ahead and see which is better, to have full screen or this way. Can you hear me, guys?
Can we have a full screen, please?
You want
Yeah, full screen is better. Yeah, if it
Okay.
might cover up something, but if so, we'll. I guess we'll
Okay, I'll do full screen here. Now, this is you know, the codes of the immune system here and how we are able to understand how the immune system works. I think I put together a very nice diagram here to show exactly the polarization of the immune system and how the, you know, the pathogens, what the macrophage eats determine exactly where the direction of the immune system is going. So if you have cells being infected virus and cancer, it goes directly to Th1, and of course you will have a lot of interferon gamma, interleukin 12, and polarizing the immune system to Th1 here, and you will have lymphocytosis, right? And when you have bacteria and fungus, you have the interleukin 6, yes, and Th17, and you will have neutrophilia, so you see it here. And if the macrophage is eating parasites or encountering pollens or other genes, the polarization is going to be Th2, and you have basophilia and eosinophilia and immunoglobulin E. And if the immune system is eating its own self-antigen, you see the polarization is moving to Tregs and then secretes interleukin 10, which suppress everything. Again, as Dr. Patel is saying, that there's a lot of variables there. It's not just strictly this. There is also something where you will see in the immune system, like multiple sclerosis, you will have both Th1 and Th17 active. And before we thought multiple sclerosis is only Th1 dominant immune disease. And then when they start producing those antibodies blockades, for interferon gamma, they find out the immune system or the multiple sclerosis flares up. And they find out it's because of the Th17 is getting stronger and stronger, and you have neutrophilia and you have destruction of the myelin sheets due to Th17. So we ultimately come up with a conclusion that multiple sclerosis is Th1, Th17, and if you want to really manage it, you need to block both the Th17 and the Th1 cytokines. You have to use both antibodies blockades that blocks the Th17, interleukin 6, and interferon gamma, interleukin 12, for really to come up with the right management for multiple sclerosis. And yes, you know, those will end up with a hybrid. That's what I'm talking about, where too much of interferon gamma can turn the Th17 into Th1. We call them X-Th17 Th1. And it's a hybrid that behave like both Th17 and Th1. And those hybrid are very difficult to suppress, even by interleukin 10. So Tregs cannot really suppress them. And if we find those cells, that's mean the prognosis of this disease is getting bad. So that's the reason I invited Cyrex company to come, the scientist. And I don't know if he's here or not, but if he's here, that would be great, because we need to detect those hybrid. I mean, I see that his lab is doing very good in detecting Th17, and he can even detect the cells, not just the cytokines. But one of the problem is that the science is progressing. I mean, we learn new things, and now we need to really come up with a way to detect those hybrid cells, because that will help us to predict the pathogenesis. And if there's any way that we can help to block or inhibit this, those hybrid, that will be very good. So the other thing that we learned from the immune system is if the immune system is moving to certain subset, let's say it's moving to Th1, then all the cytokines, which is the interferon gamma and the interleukin 12, they will be inhibiting all other subsets, all other direction. They will be inhibiting Tregs, they will be inhibiting Th2, they will be inhibiting Th17. So, but if you have the immune system is polarized into, and you have bacterial infection to Th17, then interleukin 6 and interleukin 17, they will start inhibiting all other directions of polarizations, all other subsets. And so this is how you ensure that your immune system is moving to the direction that we need to defend the body against the pathogens. And so we find out all the time when in the old school, when we learn, we find, especially when I joined Baylor College, Dr. Frank or so, it's all about Th2 and Th1. And that's what we thought. We don't know at that time there's something is called Th17. And we find out that if you have a Th1 dominant disease, immune disease, autoimmune disease, then what you need to do, you need to shift it to Th2. Okay? And if you have Th2 dominant autoimmune disease, then in order to relieve the inflammation, and we shifting it to Th1. That's an old school, but now there's other subsets called Th17 involved. So that is not enough just to shift Th1 to Th2. And when we're managing multiple sclerosis, we also need to shift the Th17 to Th2 and Th17 to Tregs. So it's a little bit different now. And when you manage multiple sclerosis, you need to shift the Th1, Th17 into Th2 Tregs. So we know that Th1 is, yes, it's mediating the cell destruction, whether it's, like I said, infected virus or cancer. And also we know that it's involved in rejecting the organs, especially organ transplant. We know that Th1 mediates the tissue rejection. Why we know that Th1, the Th2 involved in the antibodies production from immunoglobulin E to A to IgG. But in order to really make those plasma cells start producing IgG from in a state of IgE, immunoglobulin E, you need to have some active Th17 and interleukin 6. It helps to polarize the plasma cells and shift from producing immunoglobulin M into immunoglobulin G. So there's a lot of interactions of the cytokines that Th17 involved in directing which IgG that needs to be produced. What type? Is it IgG-A or E or G? So there's an interaction between Th17 cytokines and Th2 to determine the antibody types. We know that Th17, it's not bad all the way. I mean, yes, Th17 is very important to defend ourselves against fungus and bacteria. So it's very critical in our body because it helps to stimulate the neutrophils. And neutrophils does the engulfing and eating those fungus and those bacteria. But not just that, the Th17 cytokines involved in the tightening and the junction and the gut of the epithelium. So it's very critical to have those cytokines because they involve the tight junction of the gut epithelium and preventing leaky gut syndrome. But if you have overactive Th17, pathological overactive Th17 in a state of maintaining the integrity of the gut epithelium junctions, they break them up, and then you will have leaky gut syndrome. So it's very important that the activity of Th17 has to be balanced at the normal range. Once it starts to be overactive, it can cause the opposite of what it's supposed to do. So this is again and again, you know, a change in understanding of multiple sclerosis. At the beginning, we thought it is only Th1 dominant immune disease. But when we produce the blockades for the interferon gamma, we find out the multiple sclerosis flare up. And that means that there's another subset that's involved, which is Th17, that leads to neutropenia, So now, in order to manage multiple sclerosis, you need to use both antibody blockades for the Th17 cytokines and for the Th1 cytokines.
Dr. Halassa?
Yes, go ahead.
I'm thinking maybe there might be a button that can actually be nice to see you when you talk, but it's just still covering up information.
Okay.
But I think there's a button where you can actually take your
Let me see
your
if I can take
little
it
box
then.
off the screen completely.
Right, I don't know, where's that?
It might be right. I thought it would be up under the box itself. That where you.
Can you see it now? Can you see it now?
No, it's just making the screen too small to.
Okay.
I'm not sure what it looks
Okay, can
like
you see
on
the
your
box
end.
now?
The individual viewer can do that if you take your cursor and put it over the top of the screen where your picture is. On the left-hand side, there's
Oh,
a,
my picture. How about taking my picture off?
it says minimize.
Or take my picture off.
We just press the dash that says minimize, and then it won't cover up anything. Can you see it at the top, at the very top? Just run your cursor up there.
I'll just take my video off. How about that? Does that work?
No. If you take your if you take your video off, the actual lecture is small.
Okay, now, but now, can you see the whole screen or no?
No, we see you, and then the lecture is really small. It's like you switch places with the.
No, you need to. That's from your side. You need to click on that one so you come up with the lecture.
Oh, okay.
Yes. Does it work? Does that work?
Sorry, I hope I'm not
No,
messing
that's
it up for others.
No, that's okay. Any question
Okay.
up to now? Everybody is happy. Everybody is following up. I don't want people sleeping. I want you to follow up with everything we're saying here, because then I need you to expand what we learn here. This is just the basics, but I need more from your side to add more from your side. So is everything clear? Yeah, okay, great. So again, you know, when we discuss about multiple sclerosis, we find out it's Th1, Th17 dominant disease. And the reason why, how they discovered that, when they come up with the Th1 blockades, interferon gamma blockades, the multiple sclerosis flare up, and that's mean there is some other subset which Th17. And so now we know it's Th17, Th1. In order to manage it, you need to block both cytokines of Th17 and the cytokine of the Th1. Yes, you can say Th1 and Th17, they go against each other. That's true. But when you have too much of Th1, it may be those interferon gamma can really, if you cannot suppress it, you can turn it and change it. It's called neuroplasticity. You can change the Th17 or under the influence of the excessive interferon gamma, and they can become an XTH17 Th1, which is a hybrid between Th17 and Th1. And this hybrid are very resistant to be suppressed by Tregs, by interleukin 10. And then if you see those hybrid, that's mean there is a serious bad prognosis for multiple sclerosis, and we need to figure out the way that we can inhibit this hybrids with antibody blockades and with repurposing drugs. But knowing that there is some hybrids that is resistant to interleukin 10, so even if you try to shift the immune system to Treg and try to increase interleukin 10, it may not be enough to get rid from those hybrids, because they are resistant to be suppressed by interleukin 10 and Tregs. And yes, other subsets can be polarized to each other. We'll see that from the coming slide here, and even in the picture here. We call this neuroplasticity, where Th17 here, for example, acquire characteristic of Th1, and they start secreting the same thing, except interleukin 17. Plus, they're secreting even more other cytokines that leads to even more pathogenesis of the disease itself.
Is it possible for you to provide us the references for these? Because, you know, it's important for us to
Yes,
go.
I will send you references of all of this.
Okay,
and those hybrid, as you see, they are very unique. They are CD 161 CCR RS positive cells, as you see it here. They secrete interleukin-10, tumor necrotic factor, granulocyte stimulating factor, interferon gamma. They stop secreting interleukin-7, but they do behave in both ways, like Th17 and Th1, and they're very pathogenic, and it gives us bad indication of the bad prognosis of the patient with multiple sclerosis, because it's hard to really suppress them by the Treg. In addition to that, you can see some other things that can happen in multiple sclerosis, and that will aggravate the whole thing even more, especially if the Tregs start moving and becoming and acting like Th17. That's bad too, right? So that can happen as well in certain autoimmune diseases, where the Tregs, we depend on it to suppress the immune system. And because of too much of activity of Th17 in certain autoimmune diseases, it may polarize the Tregs and they behave like Th17, and that makes the autoimmune disease more worse. So those autoimmune diseases, if they are Th17 dominant and you have too much of activity Th17, they can turn the Tregs into Th17. So Tregs supposedly to suppress the Th17, preventing it from becoming wild and crazy and secreting all this neutrophil and antibodies and destroying the tissue. But if those Tregs are subject to too much of the cytokines of Th17, they can be polarized and they can become what we call the Tregs Th17. So you can see this kind of neuroplasticity and the movement and the change of those subsets to each other and become a hybrid. And that's not good. Think about this, Dr. Patel. You have cancer cells, and you know the microenvironment, they start secreting all this interleukin-10, interleukin-4, to the point, the excessive amount of interleukin-10 and transforming growth factor, interleukin-4, they can polarize the Th1 into Treg. So think about Th1 is supposed to be the cytotoxic, the immune cells that you need to activate the cytotoxic T cells to kill the cancer. And so they are the immune defense that you're depending on, right? And think about those Th1 that's surrounding the tumor. Because of excessive amount, that's the tumor microenvironment that they secrete, interleukin-10, interleukin-4, and transforming growth factor, they can turn those Th1 into Treg. So in a state of attacking the cancer, they are protecting and defending and forming a shield around the cancer. And that's a very bad prognosis for the cancers. So knowing that the cancer are able to change, what's called immunoplastic change, the immune cells from effector T cells, which is Th1, into suppressor, that will definitely cause a problem in destroying the cancer. That makes the cancer prognosis very worse. So what we do, and when we manage those cancer cells, we think, okay, let's go ahead and try to reverse that and activate those Th1 and activate those T cells, cytotoxic T cells that can also be suppressed by, as you know, the PD1 and PDL interaction, which we're going to come and talk about in the coming slides, and turn them active against the cancer again. And that may require antibodies, blockades, and repurposing drugs. Go ahead. Yes, go ahead. I'm with you. Dr. Patel, are you there?
Yeah, I wanted to bring to attention about that FOX3 and STAT3, that is the axis of developing of the either the Treg cells or the I don't know, your picture is not here. Okay.
Can you see it?
Yeah, Dr. Patel probably, it's happened to me. She probably has to adjust her own screen. No, if you go back to the other slide, Dr. Halassa, it just really quick. So the TGF beta is being created by the antigen presenting cell that stimulates the Tregs and the Th17 simultaneously.
Right. Now, so let me go ahead with that.
But I think that STAT3 and FOXP3 are also upregulated, and then there are factors that affect the FOX3 and STAT3. So I think we are just giving attention to the cytokines, but also we should look at those factors too.
Right.
Because
Yes.
those are the transcription factors.
Right. Those the tumor microenvironment do secrete interleukin-4 and 10, and this helps to polarize the naive T cells into more of Tregs, and that leads to more tolerance to the tumor. Right. Now, transforming growth factor beta, yes, if you see it, it involves an activation of Th17 and also involves an activation of the Treg. So which direction will be depend on the ratio of interleukin 6 and interleukin 6 and interleukin 10. So if you have too much of interleukin 10, then transforming growth factor will be moving the naive T cells to Treg. If you have too much of interleukin 6 with transforming growth factor beta will move the naive T cells into Th17. You got my point?
That is true, but I think that there is a factor in between. There is a one place there's a STAT3 and one place there's FOXP3. So it just depends upon who is predominant, who is not mutated, or who is mutated, and then comes in the picture all the interleukins, because I think the intermediary factor is this either the STAT3 or FOXP3. Right. And there are so many drugs which are developed, like in atopic dermatitis, they have developed so many drugs with this using the JAK inhibitor and STAT3 and STAT6. So I think that we should be also understanding the importance of those transcription factors.
Right, and that's why you really present that too, to expand what we're learning here, so we can get deeper knowledge of the inside of those cells. We're trying to give the basics now, so we can get the interest of the doctor, understand even more deeper from Dr. Patel. Yes, go ahead, Doctor.
Just a basic question here. So my understanding is that the antigen presenting cell is like, they say, for example, for the ones we were just talking about, producing the TGF beta, it's causing the naive T cells, right, to activating the FOXP3 transcription factor in the naive T cell, which causes it to turn into a Treg versus I'm just confused, like what is.
Let me make it simple, and then Dr. Patel, when she starts talking genetics in her lecture, she will be getting dig. But let's make it so easy for you guys. All the time, simplifying science is also part of the beginning of understanding, and then we go deeper. So if we have naive T cells and they are subjected to transforming growth factor, what direction they will choose, either Th17 or Treg, it depends on the ratio of interleukin 6 and interleukin 10. If you have too much of interleukin 6, then it's going to move to Th17. If you have too much of interleukin 10, it's going to move to Treg. That's a simple understanding of where they're going to move. Of course, there is deeper genetic changes that Dr. Patel is going to talk about it when she's going to present next time and expand the whole talk here. Not just Dr. Patel only, but all of you. We need to expand what we learned today so that we can come up with specific disease and use those codes to understand the pathophysiology and also how to manage it and how to modulate the immune system, and also involving the metabolic codes. You have to understand, without restoring the metabolic code, redox code, you cannot really restore the immune balance properly. And that's what the mainstream medicine especially those people they call immuno-oncologists. They say, okay, we just go ahead and give them antibodies blockades. Regardless of the redox balance, regardless of metabolic code, most of the time the efficacy of the treatment is not effective, especially if they give the antibodies naked ones without being targeted to the pathology using nanomedicine. Also, that will be a problem. Then that will trigger a lot of side effects. I think Dr. Patel is the only doctor that I know. She adapted all those protocols in her clinic, and she's using all those modalities, including redox, metabolic, and immune balance, and nanomedicine in her practice. So I really applaud Dr. Patel for doing that. Yes, so again, yes, we can talk about the Fox and how they are involved in moving the direction of the immune system, whether it's going to be Th17 or Tregs. I think Dr. Patel is going to talk about it more, but really what makes it move to this direction is the ratio between interleukin-6 and interleukin-10, which will change the genetics and expression of the naive T cells to move either to Tregs or to Th17. Again and again, we said that Th17 is not a bad guy all the way, right? It's very important Th17 to be active because it does help us to kill all those pathogens of the gut that disrupting the mucous membrane. It's very important to maintain the tight junction, but excessive activities of Th17, we have too much overactive, pathological overactive, then things can go in the opposite direction. It can lead to breaking those tight junctions. It can lead to leaky gut syndrome. And yes, you know, we thought before all this autoimmune disease is either Th1 or Th2 dominant. That's not the case anymore. There's something called Th17. Okay, so in autoimmune disease, you will find either the autoimmune disease, it's Th1, Th17 dominant, or Th2, Th17 dominant. So most of the autoimmune disease, you will see the Th17 is there, okay? And we cannot ignore it because before, when I was doing some researches in Baylor College and trying to manage allergies and trying to shift Th2 to Th1, that's all our concern is to just feed those macrophages with the allergen genes as an intracellular pathogens, which we think it will just polarize the immune system to Th1. So allergens, if it's being exposed as extracellular, the body will take them and they will polarize to Th2. So, and our research in Baylor think, okay, but if the macrophage eats the genes of the allergens, which is intracellular expression of the allergen genes, then the macrophage will be polarized, the immune system to Th1, and that's how we can manage allergies. That's what we are doing in Baylor with Dr. Frank Arsut. I was thinking, okay, he's doing direct loading the allergen genes, which is DNA plasmids from the bacteria, into the dendritic cells. I told him why we don't take those genes and load them inside the red blood cells, and then the red blood cells would take them to the macrophage and express them there and polarize the immune system of Th1 and shift the whole Th1, Th2 to Th1 and manage allergy. And that's my work that I have done with Baylor College. And then I evolved from red blood cells delivering genes into platelets delivering genes and antibodies and drugs and chemotherapy. And there's a company called Therapeutic. They're doing it, but they're using PLGA. I skipped the PLGA because I don't want to have stable drugs. I'm not selling it. I'm using just a drug, platelets, sonicated nanofilter extrusion, and coming up with simple nano particle of platelets that can be loading those drugs of interest, whether it's steroids or cancer drugs or antibiotics.
Dr. Halasa,
Yes.
so when we're talking about Treg autoimmunity, what peptide can we use to, because we know that T17, but what about Treg?
What peptides will polarize into Tregs?
No, I mean, Th17 autoimmunity, we know how to push that into the using the tier 1, but Treg cell immunity, autoimmunity, what peptide would work for that?
Okay, so let's go with the thymosin beta and alpha. And I like those two analogy here. Thymosin alpha, it does polarize the immune system into Th1, and including Th17, it polarized Th17 to Th1. So that's what's a good thing when we are attacking cancer, right?
Right, but what, not cancer.
Even
I'm
Th2,
talking about
so
the autoimmune, like rheumatoid arthritis, lupus.
yeah, so if we say the rheumatoid arthritis is Th17, Th1 dominant, right?
Right.
So what I want thymosin beta. Thymosin beta, it does polarize the immune system into Th2 Treg. You got my point?
Okay, but then there is also Treg autoimmunity. So when Treg,
What do you mean by Treg autoimmunity? There is no Tregs, they suppress the immune system. They don't do any, they're not effector cells. They don't destroy.
Here in the description here it says Th17/Treg in autoimmunity.
Yes, that if the Tregs turn and become Th17.
Right.
That's not good. Yes, that's
right. So what would you use at that time?
I still use thymosin beta to move the whole. Well, it depends now. If it's, as I said, if the disease is Th1, Th17, and the Tregs are moving to Th17 or Tregs are moving to Th1, then I would move thymosin beta, because thymosin beta will move the Th1, Th17 into Th2 Treg. You got my point? So before we thought, okay, the thymosin beta just moved the Th1 to Th2. The reality is that thymosin beta also helps to enhance the Tregs, not just Th2. So they move.
So how do we know that which autoimmune disease is Th1 dominant and which one Th17, Th1, Th17, and which one is the Th17 and Reg cell?
Right. So basically, that's where you need. We know from general that rheumatoid arthritis, most of them, they are Th17, Th1 dominant.
I have a good answer.
That's something well known. But I will just 1 second, Doctor. Okay. But still, it would be good. That's the reason we need this Cyrex company and immune science to be tested for confirming that's the case. And also when we start treating, we can see the change that's happening when we start giving those peptides and those repurposing drugs. Go ahead, Dr. Juice.
Yeah, and that's what I learned this weekend with the lecture. And I apologize. I think it's Dr.
Bojani.
Bojani, yeah. It's the lymphocyte mapping, correct? I think that's the name of it, of the test
mapping,
that measures all the different and I didn't know, Dr. Patel, by the way, your lecture was amazing, but I know that you couldn't make it, so I didn't know if you were aware of the lymphocyte mapping, or if that's something that has, something that you already were aware of or using.
No, I was aware of it, but we cannot use it in New York State.
Right.
New York State is very, very limited. So I don't know how I can use my Florida license
yet in New York, maybe.
I have a Florida license, but I don't know if I see patient in New York, I can use that.
Why? The lab does not have license in New York, not because there's a problem.
Yeah, I mean, you can, they limit us what we can do. So many times we have to, that like what, we cannot use Cyrex labs, we cannot use immunoscience lab, we cannot use Great Plains lab, we cannot use the big doctors data, we can only use their urine, and other Genova lab, we can use few things. So you just have to, you can do the urine organic acid, okay, so you become, but we cannot even order the urine amino acids and neurotransmitters.
Right, right. Well,
So
that's not cool. Because
then
right now, if you want to manage the autoimmune diseases,
and
it's different
I
from
got the
before. We just
lymphocytes.
shoot in the dark and suppress the whole thing with steroids. Now we can repurpose drugs. Now we can use antibody blockades. I think we have better tools now than before.
Right.
But without the shape and without looking at the cytokines, and now they are able to even coming up with the absolute number of those T cells, those the Tregs. So it's like CBC and differential, right? Now they have T lymphocytes and differential. So you can say how much you have percentage Tregs, how much you have of Th1, and how much you have Th2. This is critical and important, and this is the immune therapy and the immune codes that we're talking about of 2021 and maybe in the future. And I think we need to advance our tools, and we need to keep up with the frontier. And that's the whole purpose of you joining chronic disease prevention management centers. We want lectures that's all about cutting edge and frontier. We don't want to hear lectures that we know it. And it's boring for those people at the frontier to start talking about something old school. I need now the next thing. I need Dr. Patel and Joseph and all the people here to start lecturing what is beyond what we just learned, which is just the basics now. But we expect that the next lecture we're going to talk about more about genetics and more about those hybrid cells and how we can manage it. And then we need to learn about the cases that you are treating and how you monetize it using those lymphocytes mapping. The problem is now with this restriction with Dr. Patel, it's also giving me a little bit, because I depend on her a lot to come up with cases. So we need to figure out the way that she may be able to do something in Florida or Bahamas or other states where she can be more creative there, because we need Dr. Patel to be part of this movement.
I thought that they were like sending kits or something that you could draw the blood and then send it out.
But as soon as the New York State, if they come to know that that lab has done the test for the doctor of the New York State and patient living in New York State, then they have a penalty or something.
Right.
Oh,
so
my
they
goodness!
immediately say that if you are New York State, no, we don't do business with you.
Oh, I think I'm just not built for this world. That's just stupid. I'm sorry. I just voiced my opinion. That's what I can't stand. It's nonstop. We're trying to figure things out. We're trying to do the right thing, and we're constantly being punished for it. Because business is business. Well, you know what? It just doesn't work for me. And I say it because if nobody else would say it, I'm going to say it.
But Joseph, you have to question yourself whether you want to continue to practice or you want to lose your license, because they will come to your office and say,
look, but I'm not doing anything wrong. I'm
no,
helping
it's
people
not the
to
question.
get better.
Doctor, you have to comply with the regulation of the state where you're practicing.
Right.
So do you do the business with XYZ lab? And if you say yes, do you know that lab has a license to practice in New York State? I don't know. Well, doctor, before you do business with the lab, it is your responsibility to see that lab is licensed to practice, because it is your responsibility.
Right.
Well, yes, always our responsibility. You can't even meet all of the responsibilities that they throw on you, and it's not even human responsibilities. It's just nothing but runaround. The reason why I don't understand this stuff now is because I was so busy with all the other nonsense that I couldn't study and continue to learn, and it's just ridiculous. And
even with all these restrictions, I have continued my journey and my because I don't stop there, you know, I just use the clinical judgment and see. But I have seen my rheumatoid arthritis patient that who did not respond well to TA1 and her condition worsened. So I think in that case that probably she had a T17 Treg, and I should have moved on to Th2. Sometime I rotate them. So
Right.
I
And
give
that's
them.
very, very important because we thought all the time those autoimmune diseases are Th1, Th17, but some autoimmune diseases, some percentage of them, they are Th2, Th17. So you need to move them to Th1 and Treg. And yes, that's the reason having the mapping will help to clarify things, because what's written in the books not necessarily applied for all the patients. There's variations. Like psoriasis, in some articles, most of the articles, they will say psoriasis is Th1, Th17 dominant. Even they create a drug that shifts from Th1 to Th2 and Treg. But some percentage of psoriasis is actually Th2, Th17. And so that's the reason mapping the lymphocytes will make sure that what you are treating is right. And then it helps you to monetize your treatment, because after you start shifting and modulating the immune system, you can see the progress of your treatment by testing it again after maybe 2 to 3 months after you start.
So you know, I'm just wondering that the Quest lab must be doing business with Mayo or some other lab that they can do this subspecialty test. I mean,
Well, I think that's the best way of doing it because of the variability here. Like, let's say most of the ulcerative colitis, they are Th2, Th17 dominant. Most of the colitis, what we Crohn's disease is, most of it, it's Th1, Th17 dominant. And so they're both inflammatory bowel diseases, but they're dominant of the immune system, or the factors are a little bit different. And so the way that we manage it has to be also different. If it's if they are Th1, Th17 dominant, you don't give them interferon gamma, but you possibly give it for ulcerative colitis because it's Th2, Th17, most of them, not all of them. So that's the reason we need to do those mapping to make sure that we have the right polarization or the right dominant of disease and start moving it instead of suppressing the immune system and putting the patients in chaos of suppressing the immune system, putting them on risk of cancer and infection. So multiple sclerosis is chronic inflammatory disease involving destruction of myelin in central nervous system wide matter, only central system, not the peripheral. So this is a central motor central neuron disease. It's not peripheral. And this disease, what we see the patients, they have problem with both sensory and motor, because you remove the myelin sheets. So you have problem with conductive of electricity throughout the sensory and motor. They don't move fast. And you will have problem with vision, which is unilateral vision loss. You will have diplopia, you have gait problem, because in order to have gait, you have to have fast communication with the neurons to feel where your position of your legs and coordinate with movement. You cannot have a slow conduction of electricity. So myelin sheath is a very important factor, both in motor neuron and sensory neuron. Loss of bladder control, among other varied symptoms of nervous system manifestation that we know about multiple sclerosis. So it's upper motor neuron disease. Multiple sclerosis patient, we will see myelin reactive T cells. Most of the time they are Th1, Th17 dominant. And so if as I said, if things got progressed and we have Th17 by immunoplasticity turn into Th1, that is bad prognosis. Those hybrids, if you detect them in those spinal fluid, I think that is indication of the bad prognosis of the disease, where they start behaving both way, Th17, Th1.
But I want to bring to attention that that when we call about the central nervous system, in my lecture, I included that the spinal cord is part of it. And when they include when it occurs in many demyelinating foci in the spinal cord, it not necessarily it will be only motor, because patient will develop lot of sensory sensation
Yes,
because
it's
of the
not motor
involvement of
neuron
the
disease. This
sensory
is
system.
I
Right.
said upper motor neuron, upper neuron disease.
Yeah.
It's both sensory and motor. You will
Right.
lose the myelin sheath in both sensory and motor, but upper upper neuron disease, not lower neuron disease. That's the versus the lower one that we know, Guillain-Barré syndrome and all that stuff. So the pathogenic here, when you see those X, Th17, Th1, that's pathogenic, indicating the bad prognosis, those hybrid T cells. And the way that we manage multiple sclerosis, if we are doing the mapping here, is to use those antibody blockades. Yes, you can use them. That blocks the interleukin 17, interleukin 6, and interferon gamma. You have to use them all together. You cannot use just one. And also use repurposing drugs as much as you can that polarize the immune system from Th17, Th1 to Treg, Th2. And we have many of those repurposing drugs. And so again, multiple sclerosis is not anymore just Th1 dominant, it's also Th17. We can note that there's also interleukin 17 and interleukin 12, interferon gamma very high, and they all contribute in the pathogenesis of multiple sclerosis. And when we try to manage it, we need to block them both, because if you block one, it may aggravate the other side. And use those interleukin 17, interleukin 12, interleukin 6 blockades, antibodies, and also use the repurposing drugs that can also block them. We have repurposing drugs that can block the TL17, methylene blue, ivermectin. They are very powerful interleukin 17 blockade. And so we have certain FDA-approved antibody blockades that's in the market, and I think Dr. Patel is using some of them to manage chronic disease, including cancers. And I think it's adding it to platelet nanoparticle is a good idea. Injecting it freely is not a good idea because it may trigger other problems in the body. So having it targeted to pathology, it's the best way by loading inside nanoparticles, platelet nanoparticles, or using nanotechnology. Or you can use drugs. Those are TAN, is very effective. We're going to learn about that. Methylene blue, ivermectin, and other drugs that we know. Buffering the redox will help too. You know, when you buffer the redox, you restore the immune balance. So if you have too much of Th1, get it back into Th2. If you have too much of Th2, it gets back into Th1. So it's balancing really. And when you buffer the redox, it just go back to the physiological level. It does not favor much which side you move. Although some studies, they say if you have peroxynitrates as a free radical, it moves more into Th2, Th17. If you have more of a reactive oxygen species, move more to Th1. So there are some studies about the different reactive species may have different shifting. But in general, if you buffer the redox, and most of the time when you buffer the redox, you are decreasing peroxynitrates and the oxygen radicals. You are restoring the balance here. And so in order to restore the redox, you need to make sure you restore the metabolic codes or the metabolic profiles, including all the level of the hormones and all the level of your metabolized sugar, fat. All this needs to be restored back, because if you do have any imbalance in your metabolic, then that can translate into imbalance in the redox. That can be translated into imbalance in the immune system. So the mainstream medicine, they don't care about metabolism. They don't care about redox. They just go ahead and give those antibodies, and it's not going to be as much effective. So if you have diabetic patients and their metabolism is in the wacko, when you give them antibody blockades, it may not work, because those free radicals generated from the sugar and from the insulin, excessive, they may interfere with, or it may cause those shifting in the wrong direction. Holistic approach, yes, definitely. I mean, dieting is very important as well, especially with the gut. We know for the fact if you have leaky gut syndrome, what will happen? You will have too much of free radicals leaking from the gut into the body, which aggravates the underlying chronic disease and maybe cause more shifting into the Th17 and Th1. Managing leaky gut syndrome is part of managing any underlying autoimmune disease. And so if you give them those antibody blockades and you have leaky gut syndrome, you're not doing much because you still have those free radicals, that's generators that's been leaking from the gut that aggravate the shift into the wrong direction, into excessive Th17 or Th1. So making sure that you're giving the right the probiotics that will kill the pathogens, especially giving those probiotics that will help to destroy those pathogens that we know about those bacteria. It may help. The other thing, in addition to probiotics and prebiotics, which helps to clear all those and all those pathogenic microbiomes and replace them with the healthy ones that will keep the integrity of the gut epithelium. We find out also if you eat too much of salt, this can cause damage to the good bacteria gut, can destroy the good bacteria gut and lead to the bad guts to take over and lead to leaky gut syndrome. So that's the reason having less salt, it's a good idea. So we don't destroy the good gut bacteria and allow the bad gut bacteria to repopulate, which is, for example, the SFB, which can aggravate your the autoimmune diseases. The other thing.
Can I ask a quick question? Sorry, and just to while we're on it, so basically, so the Th1 and Th17 cells are overactive and stimulating the microglia cells to turn into an inflammatory or shift into an inflammatory state, and it's actually the microglia that's causing the damage to the myelin sheath.
Yes. Yes. So
okay. Those are the inflammatory cells.
What's that?
These are the inflammatory cells.
The inflammatory cells are the microglia cells,
are
being shifted into an inflammatory.
To the dendritic cells, and they are the one who cause the destruction and of the myelin sheaths. Yes.
Okay, so I do have good news. There's a peptide that I found, I don't know if you're familiar, recently called ARA290 that modulates the microglia cells. So maybe something we can, I just wanted to throw that in there.
Any peptide that helps to shift the inflammation to Treg and to Th2, that will be a treatment for multiple sclerosis. So find out exactly what shifted does, because it helps to decrease the inflammation.
But in my opinion, I think that the TB4 works really beautifully. You just have to titrate the doses. Sometimes you have to give with different routes than just the subcutaneous route. I cannot discuss too much on the phone, but I can discuss in a private discussion, but we don't know, this may go to YouTube. So, but there are different ways you can handle that, and it is still available. So you can, and it is neuroprotective. You are not, it repairs, and it is neuroprotective. If you see my slide on TB4, you will just say everything we want to do for multiple sclerosis, TB4 can help to do that, and to reduce the bacterial and spirochetes and the cascade of inflammation through the naive T cells and going into the different sequencing. You can just prevent them when you take care of patients the bacteria, spirochetes, or viruses, and then you give patient this treatment, and you're just not going to do it alone. You are going to use the redox buffer. You are going to buffer the redox and restore the immunity, you know. So it's important that you have to have a multi-pronged approach. You just cannot do just one approach. And I think that is what I really want to convey, that we are going to learn all these. We are going to learn, because this is what we are doing, is a molecular pathogenesis. We are going to learn that, and how, and it is not molecular, but it is immunomolecular. And so we are going to learn this. We are going to see what cytopathologic changes it causes. What are the, like in my lecture, I said that Th17 is the initiator and Th1 is the propagator of the disease. And that's a very, very powerful statement. And then there was another point I wanted to point out, that in the patients who died of MS, in Dr. Benz from the Europe, he did the autopsy specimen, and he looked at the exons, and he saw the spirochetes in the exons where there was a demyelination. So even when you treat that, you cannot say that, oh, well, I have treated this patient, I did that, patient got better, and now patient is in a relapse. Okay, because that means that the initiator of the inflammation is still around, and that infection is still around, whether that infection is in a form of the log filamentous spirochete, or is in the cystic fibro shape, or it is the bleb shape, and that makes them the chronic inflammatory molecule, which just keeps on stimulating the immune system, macrophages, NAPCs, and so on. So I think my impression here is that Dr. Halasa is doing a great job of explaining. But I think that when you are treating the patient, you have to corroborate everything into it, and then you have to process. And that's why sometimes it is very important to present the clinical case after in the middle of what you are presenting, and that clinical case will tell a lot to the physician how to manage it.
Right. I believe following up with clinical cases, it's very critical. So we can translate what we are learning here into more practical approach, because there's many factors that can aggravate the multiple sclerosis. Any metabolic imbalance, let's say sex hormones, leaky gut syndrome, thyroid, name it, you know, they can aggravate the immune imbalance. So fixing the metabolism and looking for all the metabolic errors, it does help, because once you fix the sex hormone, once you fix the thyroid function, and not fixing the thyroid function the way that I hear in the session of the Academy of Environmental Medicine. And I didn't kind of interrupt, they call it, but this way that I believe in my discussion here, where we have people, it's not interrupting, it's more of, you can say, discussing and trying to rectify things, so that we will not be learning something that we consider old school. We need to update ourselves, and we are not there in the setting of teaching. We're in the setting of learning from each other, because we're all doctors there. So one of the presentation that happened is that the patient has inflammatory disease, and the inflammatory disease, and when they did the metabolic panel, they find out that T3 is conversion from T4 to T3 is it's kind of abnormal. It's there's this conversion there. We have to have enough of T3 to do the function. So there's a problem with conversion of T4 to T3 from the problem with iodinase enzymes, because they are very sensitive to redox. And so if you have underground inflammatory diseases and you have too much of free radical, you can inhibit those iodinase enzyme that is responsible for converting T4 to T3. And the doctor said, well, we're going to go ahead and give them T3. What will happen if you give T3 without fixing the inflammation? The T3 will activate the sympathetic activities or sympathetic tone, and sympathetic will release the norepinephrine. Norepinephrine will stimulate the NOx enzyme, generate free radicals, and that will aggravate the whole inflammation and further suppress the conversion or inhibit the iodinase enzyme and further inhibit the conversion of T4 to T3. And so the patients, they actually feel more worse by giving them T3. What we need to do is to fix the inflammation as much as you can. And after you do all the fixing, then it's okay to say, I'll justify giving T3 if it's still the T3 is low. But most of the time, the T2 will store back to normal. But in case it got shocked or stunned by the inflammation, you can go ahead and say, I'm going to give them T3. But you have to make sure that you fix all the root cause of the inflammation before the giving T3. So that's one of the thing that I was trying to, they call it interrupt, but I call it rectifying. And I think the doctor, he came to me and he told me, you know, I appreciate you have good understanding about inflammation. And some doctors, they turn their head to me and they say, yes, we give them T3 and it's more worse. Which lines up with what we are trying to say here. The same, and why the body does that? Why the body shuts down the iodinase and prevent the T4 from T3? This is designed by the one who designed this body. It's for purpose. And why? Because when you have too much inflammation, you don't want to have too much of sympathetic. So one way the body does it is to stop the T3. So you don't have sympathetic overactivity, you don't have inflammation. So it's an anti-inflammatory effect. The same thing we have when a patient is in inflammation and they have iron deficiency, or what you call, well, we can see it, microcytic anemia. We thought it's iron deficiency, but it's a microcytic anemia, but their transferrin is high and their saturation is high and their ferritin is high, right? And they have microcytic anemia. You don't give those patients iron because they will not be absorbed, first of all, because your liver will start secreting hepcidin, which inhibit the absorption of the iron. And if you give them iron, iron will oxidize because of the inflammation that you have there and turn into free radicals, ferrics, and actually causing more inflammation and more damage to the body. So the body is protecting itself by secreting hepcidin, by preventing iron absorption. Is a protection. The same thing that inflammation, it stops that iodinase from converting T4 to T3. That's a protection. And you don't want to mess up with that protection by giving T3 or giving iron. So those are very important as an integrative doctor, as a chronic disease prevention management specialist. We learn from our mistakes. We understand that there's better solution, and managing metabolic disease is very important. Like, let's say we say no salt intake. It's not just because it kills the good bacteria and allow the bad bacteria to repopulate and lead to leaky gut syndrome, which leads to free radical generation and aggravate underlying disease. But also we find out there is a relationship between aldosterone and Th17 polarization. Aldosterone does inhibit the Th17 polarization. It inhibited interleukin 6 and it inhibited neutrophilia. And that is a good thing, right? And so if we have low salt intake, we have high aldosterone because aldosterone is trying to keep the salt reuptake, and we have less inflammation. If we eat too much of salt, then there will be suppression of aldosterone. Then you would have more flare up. You have more Th17 interleukin 6 and neutrophilia. and that will flare up the whole immune. So this is how salt is inflammatory. The same like sugar, glucose inflammatory, because those glucose in the blood, they turn into glycans, they oxidize and they become free radical generators, and they can aggravate the inflammatory diseases. And then when you have too much of sugar, also it leads to too much of insulin, and insulin itself, when it starts trying to get sugar inside, they also induce free radicals and causing redox imbalance. So sugar and insulin both cause redox imbalance and aggravate the whole immune disease. So less sugar, less salt is a good idea. And then you need to think about all other repurposing drugs that helps to polarize the immune system to Th2, Th17. We have many of them. And also you need to think about the Nmda receptor that needs to be blocked. So people will say, well, giving glutathione. Yes, glutathione is good, but too much of glutathione is not good because it will degrade and become glutamate. Glutamate amino acid is neurotoxic. So when you give glutathione, try to add NAC to it so you can recycle the glutamate of the glutathione. I think I learned this from Dr. Cheryl. She's here, and she told me about this. So we learn from each other here, okay? So also you need to look up all those NMDA receptor blockage, anything that blocks NMDA, like vitamin B12, resveratrol, CBD, anything that blocks the NMDA receptor will be another option of treating multiple sclerosis. But there's many other things that helps to buffer the redox, and you need to fix all the metabolic profile. Anything that's outside the range in the metabolism, whether it's sugar, whether it's salt, whether it is sex hormones, whether it's thyroid, all of this needs to be normalized in order to have an effective approach to manage multiple sclerosis. And very important is when you are injecting drugs, whatever it is, anti-inflammatory drugs or peptides or those immune modulators, antibody blockades, it would be good if you can load them inside nanoparticles, because it gets that drugs into the pathology, and using platelet nanoparticle is the best answer. Now, again, what would happen when you have leaky gut syndrome? The free radicals that will be leaked from the gut, they will go to the brain, and of course they will bypass the blood-brain barrier because you have the inflammation there, and it irritate the neuroglia, and then the neuroglia in a certain way, it will irritate and activate the sympathetic activities. And so you would have an overactive sympathetic, and then overactive sympathetic will release the norepinephrine in the gut, and that norepinephrine will stimulate the NOx enzyme to generate more free radicals, it trigger more inflammation, more leaky gut, more neuroglia inflammation, more sympathetic overactivity as a response to that, and more inflammation. And then you have that vicious circle that needs to be blocked. And I think the best way of blocking that is using or immune therapy, using redox therapy, using metabolic therapy, and loading all those drugs in the targeted nanoparticle. I think we can divide this lecture into 3 parts. The next part will be about COVID-19. So we can conclude now that to manage chronic diseases, we need to use all those tools that we have, and we need to have the immune lymphocytes mapping and knowing exactly the immune system and their direction and modulate it instead of suppressing it. So if you have any questions, please go ahead, raise your hand and ask, or just go ahead, take the mic.
It was a very excellent lecture, lot of information, and people, everyone will get benefited by reviewing it. And then I'm glad that you are going to have it in series of 3. So it will be really good.
Yeah,
and
give the time for
reinforcing.
processing those information. Yes, especially for the beginners here and people who's going to be joining us. At least now they know, we are trying to be the frontier and the frontier. And yes, we will try to reach out to all different levels to get them at our level of understanding, like yourself. And so we can go ahead and do even more deeper dig into managing chronic diseases.
Yeah, this was absolutely phenomenal, and obviously I expressed my frustration earlier, but I like how you were saying, okay, so then we have to do this, and okay, it's not fair in my opinion, but okay, so then we figure out a way around certain regulations. And the goal is, again, you said this before, is that we are working with the mainstream and making sure that we are following the regulations. Meanwhile, we are using the science to provide alternative therapies or adjunctive therapies to help that we know to be true, that are supported by the data, either directly or indirectly, and showing that we are presenting our cases and showing our results so that we can continue to learn and do things, the right way.
Okay, any other questions? Dr. Patel, do you have any questions?
No, thank you.
Okay, great. Any other have questions? Dr. Juliet, you there? Dr. Selvin, Dr. Toman?
Yes, I'm here.
Yeah, do you have any questions?
Oh, it's fascinating. I can't wait
Well,
for the
I'm very
COVID
impressed
part.
about your work on methylene blue and
Please
your
go
research
ahead.
that you're doing. Hopefully we can work together and do more together.
Absolutely.
Right. Any other questions? Dr. Joseph, you want to conclude the meeting?
Yeah, I mean, I think what I'd like to do from my end is to start presenting the cases on a regular basis, either ones that are already 6 months, 8 months into it. I do have some results for the patient that I treated in Michigan, a 33-year-old male who had diffuse thrombosis, dilated cardiomyopathy, ischemic stroke, secondary to COVID, and he and I'm not going to get into details, but I treated him about 5.5 months ago, approximately, and he had initially had a ejection fraction of 15%. I treated him status post 4 weeks. He was on a milrinone drip. I had given him what we call a tune-up about 3 weeks ago. And I won't get into the details, but I'm happy to say that his ejection fraction is now 48% from 15. And he's living normally. He's not on a milrinone drip. He is independently. He's working out. He's working. He can. He's living a normal life and continuing to improve. But that's where he's at now.
Well, cool. That's wonderful. We want to hear from you, Dr. Joseph, your case, because that's very applicable about our protocols that we are entertaining here. And hopefully other doctors will learn from you and do the same and present their cases as well.
Yeah, and we, hold on 1 second. I've got something cooking. I apologize.
So what are you cooking, Dr. Joseph?
Something healthy and low salt and low sugar. But actually, speaking of diet, personally, I've been making changes slowly, just getting closer to a strict plant-based diet, in working with my body. Everybody's different. Hopefully, I'd like to get into the science of diet, because I think it still seems like a confusing. Even though we know what to do, it'd be nice to gather up, do the mastermind with diet so that we actually have practical ideas on what to avoid, what to worry about. Some foods we have to know that, hey, look, these are not safe foods anymore, or maybe this was grown in a region that where they use Roundup, because we have to be extra vigilant, especially now. The electromagnetic frequencies are higher. We've got the COVID and vaccines, which are, down-regulating our ACE2 receptors, affecting our ROS system, throwing our Th1, Th2 out of balance, enhancing Th17. And there's a big fear and concern that there's going to be a lot of a surge in chronic diseases, autoimmune diseases, diseases like multiple sclerosis. I was even talking at the conference to some of those involved with the EMF frequencies, that those can have an effect on enhancing other conditions, either through autoimmunity or even allowing pathogens to have greater infectivity, such as Lyme. And as Dr. Patel spoke on, Lyme disease can lead to multiple sclerosis. It would be great to be able to have a fluency about these things more and more, and not to be, again, shooting in the dark, which is what I've always appreciated about this academy, is that we're always trying to have a clarity. Clarity is the best word I can think of to describe what we're trying to do, so that we're not pretending to know what we're doing. We actually do know what we're doing, and we can describe it logically, and we can describe the sequence of what's going on, and then through our mastermind, presenting the curveball. So what about this? Or like Dr. Patel said, what about these mixed or hybrid situations? And like you said, or eating too much salt, and that reduces aldosterone, which shifts more Th17. So there's a lot here. And I like what Dr. Bomer said a long time ago, wasn't that long ago, that it still comes back down to practice. Practice. For myself, I like to present myself as saying, look, hey, I didn't know any of this stuff. The more I know about the stuff, the more I realize I didn't know. I've got no shame in sounding like a beginner. And but Dr. Bomer always says, that's how you get good at it. You keep presenting, you keep going through these processes until it becomes normal. So everybody here has their experiences, different personalities, and different skill sets, so that we can advance ourselves and work our way through this very interesting time that we live in. And so with that,
this video, I'm going to put it on YouTube, and we're going to publish it so that they know exactly what we're doing. But the other parts, which is part 2, which will be focused on COVID, and then part 3, which will be focused on cancer, I think we're gonna keep that for the Academy of Oxidative Disorder, so they have the right for it. But at least we have something that we can share to the public and to the medical community, so they can recognize the pioneers of CDPMCS, which is Dr. Patel and Dr. Cheryl and Dr. Joseph James and Dr. Carrofield, and Dr. Juliette. Hopefully she will be joining soon. And many more of doctors that Dr. Don Pham, Dr. John Ronald, Dr. Kathleen O'Neill, those are the pioneers. Dr. Isam, Dr. Carrofield, I want to best that, and other doctors who are interested and going to be part of this. So we're going to share this video. I think this video shows the way that how we doctors, we learn from each other. We're not here to teach each other. We are here to learn from each other, to expand our knowledge, to socialize, and have that kind of spirit of advancing, without that ego and without that I know better than you, all these things that we don't want it to be part of our platform. And I believe everybody here is in the same page. We have the same chemistry. We like each other. We support each other. We back up each other. Hopefully, Dr. Klatz also is very interested in what we're doing, which is the founder of A4M. And hopefully we're working on something, maybe franchising or putting together centers of CDPMCS. So I'm working on that as well, putting a business plan for that. And then that would help us to get the resources and the money and the funds that the doctors need to expand their operation, to have their own building. Why you're renting a building? Why you don't have your own building, and invest in real estate and have other perspective, so that we can secure our future and advance medicine and make things that's, you know, more effective, efficient for the patients and for our practice. And hopefully all these protocols will be adapted, and we have a clinic that's going to be modeled. I can see Dr. Patel is almost there with all the things that she's doing. She's a very good model for reference, but I think we need to create that model that all of us on the same page on it, and all the components of our protocol is being applied, all these tools that we are learning here in our webinar. So hopefully that center is going to be coming up soon, and that will be the training center for all of us. And we can branch out and we can communicate and we can help each other by visiting each other, and because everybody has his own skill sets that is more advanced than others, and everybody has his own thing that they like to do, so we can mix things together and come up with a very nice working and communication and collaboration between all the physicians of the CDPMCS. So anyway, we'll see you next Monday. We'll possibly we're going to have 2 lectures because there's some vendors also, they want to be part of this to sponsor this activity. So we're going to give them some 15 minutes of talk. But definitely we're going to cover the second part of this lecture. You're here is very important. This is going to be discussion more than lecturing. And I'd like to see you next week, and good night, everybody, and see you then.
Awesome. Thank you.
Thank you very much for coming. Bye.
Thank you.
Thank you. Bye.